this chapter or this section of nucleic acids, I just want to do a little quick review, okay? And then I'm just going to talk about RNA, and that would be it, okay? So this is, I'm going to use this diagram as a recap, okay? This is a DNA strand. How do I know it's a DNA strand? Because I see thymine over here, okay? So I know this is a DNA strand. So first things I want you to just stress, okay, is this is what we call the sugar phosphate backbone. Okay, why? Because it's made of sugars and made of a phosphate group, and it's always going to be the same pattern: phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, or sugar, phosphate, sugar, phosphate. Okay, that's one thing. Okay, that's one thing. I'm going to erase. So feel free to pause it. Another thing I want to review really quick is how do we make this bigger? How do we make a strand bigger? You're always going to add a new nucleotide, okay, at the 3' end. So here, okay, you always, when DNA is synthesized, okay, it's always going to be synthesized in the 5' to 3' direction, okay? So DNA is synthesized, which also means made, okay, in the 5' to 3' direction. Okay, that means that nucleotides are going to be added only at the 3' end, okay, of the growing strand. So you're going to keep adding. Well, so what goes over here? You're going to have to add another phosphate group over here to be able to link to this other sugar. Okay, the 5' end. Okay, the 5' end is going to be phosphate. I'm going to do this in a different color, and the 3' end is going to have mostly. So usually it's a hydroxyl group. Okay, OH or just an O over here. Okay, so that's what it looks like, and always add the new nucleotide in the 3' end. Okay, so never add it to the 5' end. You always add it here in the 3' end. That's how you make this thing longer and longer and longer and longer. Okay, I'm gonna erase, so feel free to. Now you see how here you have the sugar phosphate backbone, right, which is here. I show you already. And then what sticks out? The nitrogenous bases. Okay, adenine, guanine, cytosine, and thymine. Okay, one more thing. I want to do this in a different color. For green, I want to do adenine and guanine. They are purine bases, right? They're purines. That means they're going to be made of 2 rings, right? Cytosine, I'm going to do this in a different color. Cytosine, thymine. Okay, these are pyrimidines. 1 ring. And eventually, what's going to happen? I'm going to do this in black. Adenine, for example, is going to pair up with another nucleotide, okay? And it's going to do some hydrogen bonding, okay? It's going to do a hydrogen bond. Let me do this in just one line, just so you can get the idea. It's going to do hydrogen bonding, right? With another nucleotide, with its complementary nucleotide over here. Okay. Let's finalize this chapter. RNA. RNA is also a nucleic acid, and A is a nucleic acid. It's a polymer of nucleotides. It has a sugar, ribose sugar, its phosphate group, and its nitrogenous base. Okay, the sugar I'm going to put here, the sugar for RNA is going to be ribose sugar. Now, what are the main differences between RNA and DNA? Well, the first difference is, and I said this already, but let's amplify this. Uracil is going to be present in RNA. Thymine is going to be present in DNA. Simple as that. Okay. So in DNA, okay, in DNA, thymine is going to be able to pair up with, guess what? With adenine, right? What about uracil? Uracil is going to do the same thing. Okay, it's just gonna replace thymine for uracil. That's it. So uracil gonna pair up with adenine. Okay, this is what you're making a new copy. Okay, this is when you're making a new copy of, for example, of RNA. I'll explain that in a second. Okay, structure. The structure of the RNA is long and it's single-stranded molecule. Make sure you know that. It's a single-stranded molecule. DNA is not single-stranded, okay? DNA is double-stranded. It means two nucleic acid chains. And remember that because that's very important. That is the reason why RNA is not our blueprint, okay? RNA is like an intermediary, okay? We have to make, technically what happens is this, the DNA is in the nucleus of the cell or in the middle of the cell. And it may be too big to be able to move from the nucleus to the cytoplasm. So it to make a copy of a little bit of that gene, okay? And that copy is going to be an RNA. The RNA is small enough, it is still long, but it's small enough to leave the nucleus, go to the cytoplasm, and being read by a ribosome. So that's why RNA is going to be single-stranded. It's simple, it's smaller, okay? DNA is going to be double-stranded. The types of sugar. I already said this. RNA contains ribose. DNA contains deoxyribose, and there's only one tiny little difference with the sugar. Okay. And finally, the function. What's the function of DNA? Storing genetic information. It's going to determine the order of your amino acids. It's going to be your blueprint. What about RNA? RNA is just going to carry genetic information to the ribosomes. So like I said, it's like an intermediary, right? It's like a middleman or middlewoman, whatever you call it nowadays. Here in this picture, you can see those changes, right? This is a nucleotide it's a nucleotide in RNA. Nitrogenous bases, you have adenine, guanine, tyrosine, no thymine, but you have uracil, right? And this is going to be uracil. What about the sugar? The sugar is going to be ribose. Okay, so I have this one over here. Okay, no H over there instead of a hydrogen. So this is the sugar of RNA. And then finally the phosphate group, which is going to be very similar to the phosphate group in the DNA molecule. Last but not least, DNA. Like I said, DNA is double-stranded. Okay, it's going to have one strand going one direction, and the other one is going to go the other direction. Okay, maybe it's going to be one goes this way, the other one goes the other way around. Okay, that's what it means. That's what it means when you hear that both strands of DNA run anti-parallel to each other. Okay, one goes 3' to 5', the other one 5' to 3'. Okay, so one goes one direction, the other one goes the other direction. Okay, and then you have your base pairs over here, your base pairs here. Okay, RNA is going to be single-stranded. Okay, that's it. You see how there's not another one over here. The reason why there's not is because RNA is a single-stranded molecule. And that, ladies and gentlemen, is the end of our chapters, the end of our nucleic acid section. I hope you learned something. Next chapter will be about the cell.